very happy to talk to you about our recent work on the use of repetitive TMS to treat cannabis use disorder in people with schizophrenia. This work was conducted at CAMH um, with the support of a grant from the National Institutes of Drug Abuse from the US. I also give my disclosures on the second slide. Just as a bit of background, many clinicians know that people with schizophrenia use alcohol and drugs at much higher rates than in the general population. And in fact, for cannabis use disorder, the rates are around 25% as opposed to 3% in the general population. We know that cannabis use is associated with more frequent hospitalizations, a worse course of the disorder, early onset of schizophrenia, and also decreases in cognitive function and psychotic symptom stability. We also know that, at least in terms of the pathophysiology of the disorder, that there is evidence for a putative dysfunction of the dorsolateral prefrontal cortex. And in fact, the figure on slide 3 is from a published review article from my group that talks about the various interactive factors, including genetics, neurotransmitter abnormalities, and the effects of the environment that ultimately lead to the initiation and maintenance of cannabis use behaviors in people with schizophrenia. The next slide shows a preparatory study that we did using extended cannabis abstinence in people with schizophrenia, not only to potentially treat cannabis use disorder in these patients, but to understand the effects of cannabis by prospectively taking off their cannabis use using a contingent reinforcement abstinence paradigm. And with this single $300 contingent payment at the end of 28 weeks of treatment, we're actually able to get about 42% of patients and just over 50% of controls who had cannabis use disorder to quit at the end of that 28-day period. So this was very exciting news for us. The picture is of my former graduate student, Rachel Rabin, who's now an assistant professor in the psychiatry department at McGill University in Montreal. And Rachel did a series of programmatic studies during her PhD training with me that showed that not only could we achieve high rates of abstinence, but that there were selective improvements in cognition, reductions in depressive symptoms, and an increase in a compensatory manner in tobacco smoking during 28 days of cannabis abstinence. Having said that about treatment, there are no approved medications for treating cannabis use disorder, and behavioral treatments for the most part, including cognitive behavioral therapy, coping skills, motivational interviewing, and the few trials of contingency management have shown only modest effectiveness. The inset is a graph showing the improvement by about 40% in verbal learning and memory on the Hopkins Verbal Learning Test during a 28-day period. And then when we took away the reinforcer, most of those people relapsed, and with that came a further worsening of performance on this verbal learning and memory task, known as the Hopkins Verbal Learning Task. And this work was published a few years ago in the journal Neuropsychopharmacology. So our question, knowing that the prefrontal cortex is a potential target for treatment development, and if alters cognition and potentially also regulates some of the reward centers in the inner core of the midbrain, where the nucleus accumbens and the ventral tegmental area are located, we wanted to know if a treatment targeting the DLPFC could improve cognition and reduce cannabis use levels in people with schizophrenia. So we used repetitive transcranial magnetic stimulation. And this is effectively weak magnetic pulses that are applied to various parts of the brain. And in our case, this was bilaterally to the frontal lobes, to the dorsolateral PFC. which is known to be important in judgment, planning, and executive function. Now, we used a high frequency stimulation procedure that has been developed at CAMH and in Toronto over the last 20 years. In fact, this 20 hertz treatment 5 days a week for 4 weeks has been shown to improve cognitive control, including NBAC and spatial working memory. And we figured that applying this to the DLPFC could lead to better outcomes in substance use disorders. And we actually had a paper on this, a review of brain stimulation methods to treat substance use disorders, led by a research coordinator in my lab, Alex Coles, that essentially showed that at least for rTMS and another form of brain stimulation called transcranial direct current stimulation, that in treating tobacco and stimulant and alcohol use disorders, that application of these brain stimulation technologies to the dorsolateral prefrontal cortex led to the best treatment outcomes. We have done and others have done a series of studies in people with cannabis disorder. Actually, the only 2 published studies are from a group in South Carolina led by Dr. Salem. And these were very small studies with sample sizes of between 3 and 14 patients. They were very preliminary and gave mixed effects in terms of reducing cannabis use and craving. However, we decided to use a paradigm that we have developed for our smoking studies, both in clinical trials and laboratory studies. We've done both short-term stimulations for 3 days with both sham and active stimulations, and for longer periods up to 4 weeks. And more is better. So we actually saw significant decreases in craving with the longer treatment paradigm. The shorter treatment paradigm actually did not affect craving, tobacco use, or cognition. So what we reasoned is if we could use rTMS to the frontal lobes, to the DLPFC, maybe we could have better treatment outcomes in terms of reductions in cannabis use, possibly abstinence, and also improvements in cognition. So our study sought to determine the effects of 4 weeks of rTMS directed to the frontal lobes on cannabis use outcomes in people with schizophrenia and cannabis use disorder. That was the primary outcome. And we also wanted to look at effects of rTMS on cannabis withdrawal and craving, on psychiatric symptoms, including the positive and negative symptoms of schizophrenia, and on cognitive functioning in these patients. A little bit about the study design and methods. So this is the team of investigators, my PhD student, Carolina Kozak, master's student, Darby Lowe, and 2 research coordinators in my lab, Alex Coles and Julia Sesiatic. And essentially these were men or women between 18 and 55 having the diagnosis of schizophrenia and cannabis use disorder, using at least a gram a day, and having a cannabis use disorders identification test score of 12 or higher, having a positive urine for THC, being either tobacco, cigarette smokers or non-smokers, and wanting to quit cannabis or change their cannabis use within the next 30 days. So that are the inclusion criteria. Exclusions included other substance use disorders besides cannabis, caffeine, or nicotine, any evidence of a head injury with loss of consciousness greater than 5 minutes, any other neurological or medical illness such as a seizure disorder or first-degree relative with seizures, or any other medical problems that the PI or the investigators thought was problematic. Also, we excluded people with metallic implants in their heads, a history of previous rTMS treatment, and also women who were pregnant or planning to become pregnant. The rTMS parameters are outlined in this slide. We did 30-minute treatments a day, 5 times a week for 4 weeks, and we were lucky to collaborate with a team of investigators at our Temerty Center for Therapeutic Brain Interventions, led by Doctors Daniel Bloomberger and Jeff Daskalakis. We targeted the bilateral prefrontal cortex, which was localized by EEG using the F5 and 6 electrode leads. One thing that was very novel about these study is that we used the MagPro 100 stimulator, which was equipped with a B65 active and sham rTMS coil. And the sham coil looks and sounds like the active coil, but doesn't deliver the magnetic field. And the rTMS was delivered at an intensity of 90% of the resting motor threshold at the frequency of 20 hertz, and this included 25 trains, 30 pulses per train, and a 30-second intertrain interval for a total of 30 minutes per session. The next slide shows the design of this randomized double-blind sham-controlled trial in our outpatients with schizophrenia and cannabis use disorder. So after a baseline where we determined that they were using cannabis at sufficient levels, they had cognitive testing as a training session, and then were randomized either to an active or the sham arms of treatment. In order to ensure compliance with the rTMS procedures, we did escalating payments using contingency management, contingency reinforcement at each rTMS session, and gave progressive payments to individuals such that they could make $300 for attending all the rTMS sessions. We did a number of clinical substance abuse and cognitive measures. We collected urines for urine toxicology for both semi-quantitative and quantitative assays twice a week for the 4 weeks, and also did a 1-month follow-up at day 56. We had a number of clinical and substance abuse outcomes that we measured, including the PANSS for positive and negative symptoms of schizophrenia, the Calgary Depression Scale to tease out depressive symptoms versus negative symptoms or extrapyramidal symptoms in these patients. And we also had a number of cannabis-related measures, including timeline followback, which is self-report of cannabis use, craving and withdrawal, measured using validated scales, and 3 types of different urine toxicology. For screening, we used the qualitative MedTox system. For semi-quantitative outcomes, which was our primary biochemical verification, we used the NarcoCheck semi-quantitative system. And then the definitive measure we used was sequential gas chromatography and mass spectroscopy to measure the THC metabolite carboxy-THC. We also measured a whole bunch of different cognitive variables in this study. I won't go into them in great detail. Our primary outcomes based on our previous studies were verbal learning and memory with the Hopkins verbal learning test, the revised version, the spatial delayed response, which measures visual spatial working memory, and looking at processing speed, which is known to be impaired by cannabis, using the Trails Making Test Type A. We looked at a whole bunch of other executive functioning motivational tests, and we also looked at an electrophysiologic test called mismatch negativity. However, I won't be able to show you most of those outcomes. Our data analysis, we're very interested in comparing demographic measures, so I'll present some of those data. We looked at treatment retention with classic survival curve analysis using Kaplan-Meier survival curves and statistics. Our primary outcomes for the main dependent measures of craving, cognition, and cannabis use were analyzed using linear mixed effect models, or LMs. We looked at the primary outcomes of cannabis use and secondary outcomes of craving, withdrawal, psychiatric symptoms, and neuropsychological and neurophysiologic outcomes. And when we saw evidence of changes in cannabis versus sham groups, we employed something known as linear contrast tests across primary analysis, and in all testing looked at treatment time and treatment by time interactions. Okay, so the results. I show you the consort diagram here. Ultimately, we screened 99 subjects, had 53 assessed in person for eligibility, had 24 people actually enrolled in the study and completed the cognitive training baseline. But as you can see in the consort flow diagram, a few people dropped out just before randomization. So we were left with a sample of 19 people who were actually randomized in the trial, 9 to the active rTMS group and 10 to the sham group, and had a few dropouts, but very few along the way, and a few more dropouts at the follow-up. Just in terms of baseline demographics, the populations were very comparable. It was mostly men. In fact, there was only one woman enrolled. They had more than a high school education. They actually had higher than average IQs on the WTAR, age in their early to mid 30s, diagnoses primarily of schizophrenia, some schizoaffective, fairly low symptoms consistent with low levels of psychotic symptomatology on the various PAN subscales, relatively low levels of depression with the Calgary scale, and low levels of suicidality with the Columbia Suicide Rating Scale. In terms of substance abuse, they used just under a gram a day of cannabis. They had substantial levels of carboxy THC as measured by both the semi-quantitative narco check and the quantitative GCMS-MS methods. They had very severe or moderate to severe cannabis use disorder based on DSM-5 criteria and on CUDIT scores. These people had tried to quit before, were fairly regular cannabis users. About 20% rolled their cannabis with tobacco. Most of them were tobacco smokers, smoking about 7 to 8 cigarettes per day with a low Fagerstrom test for nicotine dependence score, used very little alcohol, and had very low audit alcohol use disorder identification test scores. So first outcome I show you here is treatment retention between the active and sham groups. And what you can see here is that we had very high rates of treatment retention, 90% overall in the whole sample of 19, and 100% retention through the end of 4 weeks in the active group, and about 80% retention in the sham group. In fact, 2 subjects dropped out in the sham group. But when we analyzed this using the log rank test statistics that's used in Kaplan-Meier analysis, this obviously was not significant. Okay, so our primary outcome measure was cannabis use between the active and sham groups. And what you see on the left-hand side are the self-report on the top panel and the narco-check semi-quantitative toxicology in the bottom panel. And what you see, at least qualitatively, is a clear separation between the active and the sham group, such that in the active group there seemed to be more of a reduction, both based on self-report and narco-check. When we did a little bit more fine-grained analysis of these, we discerned that there was medium effect sizes, Cohen's d effects from baseline to endpoint of about 0.5 to 0.72. And that was also shown with the semi-quantitative analysis. So the key message here is that active versus sham rTMS decreased cannabis use over times with greater reductions in the active versus sham group. However, based on the low sample, we did not reach statistical significance. The next slide looks at one of the primary secondary outcomes, and this was cannabis craving. And what you see here, using the primary linear contrast test and treatment by time interactions, is an almost significant treatment effect of active rTMS in reducing craving over the 4 weeks, and effectively no change in the sham condition. We also looked at symptoms of schizophrenia, primarily positive and negative symptoms. And what we show here are positive symptoms, which seem to show the greatest effect, and active rTMS at 20 hertz, producing about a 10 to 15% reduction in positive symptoms, but no change in the sham group, and a nearly significant treatment by time interaction and a significant linear contrast test. We looked at a whole bunch of different measures of cognition, including some of the primary measures, but essentially found no effects, at least in this limited sample, except for looking at an attentional measure on the continuous performance test, that is hit rate reaction time. And what we saw was a slight reduction, about 5 to 10% in the active condition, and actually an impairment in reaction time in the sham condition. So there seemed to be fairly selective improvements of cognition confined to sustained attention on the CPT test. Finally, I show you outcomes related to a compensatory increase that we've seen in tobacco use with verified cannabis abstinence. Now remember, most of these people only reduced their cannabis use, but what we were able to show was that active rTMS could suppress the increase in tobacco use over the 4-week period, whereas in the sham condition, we see ultimately what looked like a compensatory increase during reductions in cannabis use in the sham condition. Just to round things out, this treatment was well tolerated and safe in terms of adverse outcomes. Really no differences statistically or clinically between the groups, suggesting the safety and tolerability of high-frequency rTMS in cannabis using people with schizophrenia. Okay, so in conclusion, we tried to exploit the known pathophysiology of schizophrenia and co-occurring cannabis use disorder by targeting the frontal lobes, and presumably the effects we saw were related to an increase in cortical excitability and activation of synaptic plasticity, which led, at least selectively, to improvements in cognition, and perhaps a top-down modulating effect on the reward pathways based on stimulation of the prefrontal cortex, which would reduce subcortical, dopaminergic, and other reward center function, reducing craving, and ultimately reducing cannabis use in the active rTMS condition. Some strengths and limitations of our study. This was a prospective trial using a double-blind, sham-controlled, randomized design. We used a well-validated rTMS treatment paradigm with rigorous screening of subjects, well-matched treatment groups, cannabis use monitored by self-report and by 2 objective measures of urine toxicology testing, both semi-quantitative and quantitative testing. We did progressive payments to ensure that treatment retention was high. However, there were some significant limitations. We were statistically underpowered to look at differences in the trial. The majority of the sample was male, therefore reducing the generalizability of our findings. And rTMS is no easy thing to employ, and so the feasibility of rTMS for this population, given the difficulties of recruiting, treating, and retaining these people with such a time-intensive neuromodulation technique, we have to continue to revisit that. Okay, so in conclusion, there is promising therapeutic potential for high-frequency rTMS using a 5 times a week paradigm for 4 weeks in reducing cannabis use and craving, improving positive symptoms, selectively improving some aspects of attentional functioning, and preventing a compensatory tobacco use increase in people with schizophrenia in the active versus sham conditions. Of course, given this small sample size, we'd like to do a more definitive study, probably using a multi-site treatment paradigm. We did a reverse power calculation, which showed that based on our self-report data, we need a total of 78 people in this study, or 39 per group, so about 80 people. And we're actually in the midst of planning a multi-site study with 3 other centers across the United States and Canada. We also want to look at mediation models, and one of my students who was part of this project, Darby Lowe, completed her master's degree looking at the mediating effects of cognition between rTMS and its effects on cannabis use, and actually found some interesting findings that we're doing more fine-grained analysis on. Finally, the use of more deep, non-invasive methods like the H-coil or deep rTMS to target associated reward structures like the amygdala, the insula, and the nucleus accumbens may be worth um, uh, looking at in future studies to treat cannabis use in people with schizophrenia. I'd just like to thank our funders, the National Institutes of Health, specifically National Institutes on Drug Abuse, through a grant support to me over the last several years, our support for our preliminary studies from CIHR, of course, support from our organization, CAMH, and the Institute of Medical Sciences at the University of Toronto. I'd also like to thank my students, Dr. Carolina Budzinski and Ms. Darby Lowe, who both graduated within the last year with their doctorate and master's degree. My collaborators, which included Dr. Michael Kiang, who was formerly at McMaster, Dr. Daniel Bloomberger from our Temerity Brain Stimulation Center, and Dr. Isabel Ballou, a neuroscientist at CAMH, Dr. Gary Remington, an expert in schizophrenia, Mr. Marco Sanchez, our biostatistician at CAMH, and also Dr. Peter Jacoby, who's a brain stimulation expert at Sunnybrook, as well as my former student, Dr. Rachel Rabin at McGill, as well as a whole bunch of research staff in my lab and in our division of substance abuse and addictions at CAMH. So I will be happy to stop there, and should there be any questions, we can arrange for me to answer that. Thank you so much. Bye-bye.